Hi there, my name is Marcelo. I'm going to present the state of art and systematics of Inuleae. I'm a federal, I will start to sharing, fully share my screen. I'm a postdoc at Federal University of Uberlândia and an associate researcher at University of Campinas. Thanks again for Bruce Weldring, for Mauricio Bonifacino and Gustavo for inviting me for this workshop at TICA workshop at Botany conference. So this presentation of Inuleae, systematics and evolution of Inuleae, is divided in three main topics: a general presentation of the tribe, a brief classification history, and characterization of the tribe. So Inuleae is a tribe of Asteraceae with 66 genera and 690 species. Inuleae has almost a cosmopolitan distribution due to the largest distribution of Plukia, but the centers of diversity of the tribe are placed in Asia, Africa, and Europe, and the Mediterranean region is certainly a hotspot of diversity for the tribe. Inuleae is divided in two subtribes, Inuleae and Plukeini. And Plukeini has its diversity more centered in southern hemisphere, with which has three centers of diversity in Australasia and Africa and South America. And Inuleae has a different pattern. It is more diverse and specials in northern hemisphere. Here I brought three images, three photographs of different genera of Inuleae to start to show the morphological diversity of the tribe. Here we have Stenacanium, Chiliadenus, and Tessaria. The most species-rich genera of Inuleae are Blumia, with 100 species. Blumia, it is placed in Inuleae. Inula is other genera with very special genera with 80 species. It's Pulicaria is also a member of Inuleae. It has 85 species, and Plukia is the most diverse genus of Plukeini. So Inuleae is placed on asteroid subfamily of Asteraceae, here in pink, purple. It is the sister group of the Eleanthe Alliance, so it's a derived tribe within the family. Here I brought an image of other genus. Cratistylis is also a Plukeini genus. And now I will discuss a bit, very briefly, the main changes in the circumscription of the tribe. Cassini established the Inuleae with 9 subtribes, and after almost 50 years later, Bent and Hoffman they follow the classification of Cassini, but they expanded a bit the circumscription, including some genera, some recently described genera, changed some species from genera with other tribes and brought to Inuleae. And Max Muller, almost a century later, made the first reevaluation of the works, the works of classification of Hoffman and Bentham, and they used as source of evidence morphology, cytological information, as well as phytochemical information, and they uh, proposed that Inuleae should be reorganized in three subtribes: Inuleae, Antrixini, and Gnaphalini. But even Max Muller and collaborators uh, discussed that it was difficult to find strong characters to circumscribe the tribe. And also these three subtribes were quite different from each other. So uh, um, they point out that maybe it need to be changed. Almost 10 years later, Underberg made a morphological cladistic analysis of Inuleae, and he found that the tribe was paraphyletic, so he proposed to segregate Inuleae sensu Max Muller in three different tribes. In Inuleae sensu stricto, or more restricted, he reinstated Gnaphalli and proposed a new tribe, Pluki, to the members of Inuleae, similar to, more related with Plukia. So after that, there were several phylogenetic studies based on morphology, and also most of it from molecular sequences that show that Pluki and Inuleae were related, and also some works have shown that Pluki was inserted within Inuleae, and with some genera of Inuleae within Pluki. So in 2007, Underberg and Eldenas decided to broaden the circumscription of Inuleae and bring back the taxa from Pluki. So they included it as a subtribe as Plukeini within Inuleae. So the circumscription of the tribe is the same from 2007 and 2007, and this is what we accept now. Here I'm showing the most important segregated taxa from Inuleae and its current circumscription, with Inuleae subtribe and Plukeini. Here, Gnaphalli was a very diverse tribe, which was segregated from Inuleae. Atroisme and Fedi, two small tribes that were more recently segregated from Inuleae. This is the picture of the main picture of the classification of Inuleae. But okay, how do I know if this specimen it's a member of Inuleae or not. But there are some characters that are useful for circumscribe the tribe. The first of it is the style lobes, the obtuse style lobes, and also each style lobe has two stigmatic surfaces, which are separate at the base and confluent at the apex. Additionally, the caudate anthers are very useful for circumscription. Regarding the differentiation of Gnaphali, Inuleae do not have fibers on the phloem and also do not have latissiferous, but it has resiniferous ducts and glands. Here I'm showing in some resiniferous glands on the fruit or on a cypselum of asterate of Inuleae. Additionally, the pollen grains of Inuleae are echinate, microperforate, and also it is cavate. So this is the most important characters to circumscribe the tribe. But there is some variation. There is some interesting variation on also on the corolla to differentiate within the tribe. Here we have the tubulose corolla. We have also this filiform corolla and the ligulate corolla. So there is some variation within the tribe, as well as in the style for bisexual flowers, mostly from with tubulose corolla. They have these short branched style arms, and in the filiform female corolla, there is this long branch style arms, and for the functionally male flowers, the style lobes, the style arms do not open; they still close, and the fruits do not develop. Okay, and additionally, the capitula. There is some interesting variation on the capitula. There is this deciform capitula, what we found in Plukia, and this radiated capitula. These are the two most important, but also there are some genera with discoid, only with tubulose flowers on, and discoid capitula with only tubulose flowers. And also the inflorescences are quite interesting because there is this type of inflorescence with isolated capitula, but also there are these specifiform inflorescence or glomerule where the capitula are aggregated, and the inflorescences, the distribution of the capitula or even disaggregated capitula are organized in several ways, from racemiform to paniculiform or corymbiform inflorescences. And okay, we can also use this morphological variation within the tribe to try to segregate the subtribes which was tried in the past was proposed to separate the tribes, and some of the characters, I will show some of the characters. One of those is the position of the sweeping hairs. For Inuleae, the sweeping hairs are placed just above the branches. But for Pluceiinae, the sweeping hairs reach below the bifurcation. Additionally, for Inuleae, the apex of the sweeping hairs are acute. In Pluceiinae, the apex of the sweeping hairs are obtuse. In Inuleae, there are most of radiated capitula with ligulate flowers and tubulose flowers. But in Pluceiinae, there are mostly deciform capitulas with a few tubulose flowers and several from almost 100 filiform flowers. Nylinder and Underberg made a reconstruction of ancestral characters for these traits that were used to circumscribe the subtribes Inuleae and Pluceiinae. And what they found was the, the distribution of sweeping hairs, as well as the apex of the sweeping hairs, were not that good characters to circumscribe both subtribes, because mostly Inuleae is more homogeneous, but Pluceiinae it's very heterogeneous with several reversals to a plesiomorphic state. So there is more polymorphisms on the Pluceiinae. And also for the shape of the marginal flowers, where most of the Inuleae has ray flowers, but also it's what they found was there are some filiform flowers on Inuleae, and a kind of reversals of kind of mass or within Pluceiinae, with ray flowers, filiform flowers, and also with tubulose flowers. As well as the color of the flower, which was important for Inuleae, but also in Pluceiinae there is more variation from yellow flowers to white, pink, or purple or blue flowers. But what they found was that one character, one trait was quite good for circumscribe the subtribes Inuleae and Pluceiinae. Inuleae has these elongated crystals on epidermic cells of the fruit, and Pluceiinae do not have this character. So this is quite good character. There is just one exception. This is Caeswalia, because Caeswalia is a member of Inuleae, but it lacks epidermic cells on the fruit. They get lost. So there is no cells to have crystals. So this is the exception of Inuleae. but this is a quite good character to circumscribe Plukeini and Inuleae. So most of the selected characters to circumscribe the subtribes were not that good. Most of it because Plukeini has several reversions to plesiomorphic states, and Plukeini is more polymorphic. So I would like to dedicate this presentation to my both past advisors, Dr. Arne Anderberg from Swedish Museum of Natural History and Professor João Semir from University of Campinas, who advised me during my PhD. And this presentation I dedicate to them. These are the bibliography that I used to build this presentation. So I will start to share my screen and also record